Hey, good morning to everybody. Thank you for being in person and to our attendees who are joining us virtually. My name is Jan. I am a faculty at BIDMC, and I'm also the director of the detection unit as part of one of our precision RNA medicine core. This morning, we are hosting a RNA Institute of Medicine RNA technology showcase, and we are company, Mirxes, is doing a presentation with us this morning. So it's going to be the business manager will have a few slides, and then Dr. Kumar will present for about 40 minutes, and we'll take questions the last 10 minutes. If you have any questions during the talk, please just type it in the Q&A box that's on your screen. And so I will hand now to Craig.
Thank you, Jan. I know you're not supposed to start a presentation with a caveat, but both Vikrant and I, if we look a little shabby, we're wearing the same clothes as yesterday morning. So our luggage is somewhere in the Northeast, and I have to find it. But thank you for the opportunity to present to the group here. I'm Craig Morley. I'm the business director for Mirxes in the Northeast. Just got a few slides about the company. Also, just as aware, we have a sheet that if you want a copy, a link to the presentation, I think, Jan, we might be sending it out, I'm not sure. But we also have, we can give you a link for the copy of the presentation and then also the publications. So Dr. Kumar's got a lot of publications of the applications of where Stereo-seq is used. And if you want to get a copy of those, they're available through that link. So please sign in on there. A little bit, just a couple slides about the company. So Mirxes was founded in 2014. It was a spin out from the Singapore Agency for Science, Technology, and and Research, A-STAR. It's in the, if you're familiar with Singapore, it's in the Biopolis research part. We're a diversified global leader in discovery genomics, both products and services. Deep expertise in genomics, and as you'll see here, especially with Stereo-seq, methylation, we have a strong bioinformatics group, and Vikrant is from the bioinformatics team. We have state-of-the-art facilities in Singapore. We have genomics for research services. We have labs with a little more quality management systems for pharma clients, and then we also do clinical genomics, mainly for Singapore, Philippines, Malaysia, in the region. So we have a partnership and we do clinical genomics as well there. So we cover a good gambit from research to clinical in the area of genomics. Since 2014, we've raised about over $130 million. And then was just announced here a few months ago, we're a private company now, we're gonna go public, and we have an IPO in the next couple of months. So I just mentioned that, just that we've been around for a while. We're a very stable company, and with a lot of good research. As mentioned, we're the corporate office and the labs are based in Singapore. We do business all over Southeast Asia. So we have facilities in Philippines, commercial, specialty clinical testing, covers all the way around Southeast Asia, Japan, and then we also have facilities in China. And we have a small proof of concept lab here in North Carolina. So what we're going to talk about today is our service arm. And so what happens is clients that want to do sequencing with us or other capabilities work with Jan, and then the samples go to our Durham facility. Then we take chain of custody, we repackage it and send it to Singapore. So we worry about customs and get everything from the US to Singapore. And then we're kind of a one-stop genomics solution on the services side. So whole genome, whole exome, RNA-seq, whole genome methylation sequencing, cell-free whole genome. Really kind of talk about today is our specialty in our single-cell specialty, RNA sequencing, whole genome, but Vikrant will really focus in on spatial transcriptomics and our Stereo-seq capabilities on that. We have multiple sequencing platforms. So we have the Illumina platform, and then we also have complete genomics technology as well. So we have the G400, T7, and then the T10, which is one of the largest high throughput systems. There's about 5 worldwide on the T10. So We do a lot of population genomics, so if you have a lot of sequencing, we would put on a T10 on that. And then also, we're the first and only certified service provider for Stereo-seq. So as a service provider, so basically the companies we work closely with them. They test us out, our performance, they feel comfortable. So they certified us as a service provider for Stereo-seq. Okay, Dr. Jen, thank you very much.
Thank you, Greg. So this morning gives me great pleasure to welcome Dr. Kumar, who flew in from Singapore. Dr. Kumar is the head of bioinformatics in the genomics arm of Mirxes. He did his PhD at the Indian Statistical Institute, and then he did his postdoc at Duke NUS, which is the National University of Singapore. And then he also did another postdoc at the Genome Institute of Singapore. He has a lot of experience with population genetics and omics data. And so let's welcome Dr. Kumar to present his spatial transcriptomics talk today.
Thank you, Jan, for my introduction and inviting me here for this talk. Thank you, folks, for coming for this presentation. So I'm going to talk about spatially resolved sub-cellular transcriptomics and quite a bit of its application. I focus more on the Stereo-seq, which is a kind of truly sub-cellular transcriptomic way of doing things. I'll focus first a little bit on the introduction and the workflow, a bit of it, and what are the advantages of using the Stereo-seq, and then finally I will focus much more on the application side. The question is, why do we do spatial transcriptomics? And if you look at the cartoon here, this famous portrait, and you have to do a bulk kind of sequencing for this, you can't figure out anything in the sense that what kind of cell types there are and where they're coming from and how they are spatially located. Then from bulk, people started moving to single cell transcriptomics. I'm sure most of you guys know that you can figure out the cell types, but then still you miss a lot of spatial context. And we all know that if you do not have a spatial context, cells do have much more meaning when they are in a spatial context. And then came some of those spatial transcriptomics, which were low resolution, but still the picture is not very clear, not sharp. And then comes the high resolution spatial transcriptomics. And that's where I think we're pitching in Stereo-seq. In terms of development of spatial technologies, it was there quite a bit long back, started in '82 with small molecule fish. And then over a period of time, it became multi-probe, different technology came in. And in 2021, Stereo-seq came, which was essentially in situ capture technology. I will go into this a bit more. And of course, there were other kind of technologies like microdissection gene expression technology, which was very famous for a while, LCM. And then you had, of course, a lot of them are in situ hybridization technology as well, like Mirxes and all. Now, Stereo-seq, what's that? It's a spatial enhanced resolution omics sequencing. It doesn't require any kind of a machine or anything except for a chip. Now you have this chip here. Let's say this is a 1 centimeter or 1 centimeter chip, and it has millions of spots. Each spot is the size of 220 nanometer, and the distance between two spots is around 500 nanometer to 700 nanometers. So what this spot contains? It contains DNA nanoballs. Each DNA nanoball in this, each spot contains a lot of DNA nanoball, which comprises of poly T, so which essentially this is a poly A capture technology. It has UMI so that you can remove the deduplicated RNAs or transcripts. And then it has CID, which is essentially X and Y coordinates for each of the spots. So each spot can be picked up by the CID. So what you do is you take a tissue, you section the tissue, you mount that tissue here in this kit, and you permalize that. Once the permalization happens, The poly T will capture all those poly A. Then you make cDNA out of it, and then you do library and sequencing. And that's pretty much is done here. And once the sequencing is done, you get a data set which can be used for any kind of algorithms available here for single cell and even spatial algorithms. Now, just briefly about the workflow. This is first workflow, which is quite a bit of cheap workflow in the sense that we don't go to the second workflow unless until this first workflow is clear, which is primarily a permeabilization workflow. You take the tissue, you mount that tissue, and then you test for permeabilization testing at different time points. We usually do 3, sometimes we can do 4 as well. And then we try to figure out what is the best permeabilization time, what is the most optimal permeabilization time. Once that is figured out here, then we take another section and then do the whole workflow, which is experimental preparation of the tissue. We mount the tissue, we fix the tissue, we take the image, which is very important, taking the image of the tissue. And then based on the F1 workflow permeabilization, we use that permeabilization timing here. And then we do reverse transcription to create cDNA library here. And we collect the cDNA, we purify it, we create a sequencing library, and we sequence it. And that's pretty much what we do here. But mind you, the permeabilization is a very important factor here. The much better it is, a lot of cDNA you will get. Once you get the sequencing data, which essentially is FASTQ files. Now, these FASTQ files will require other 3 kinds of files as well. One is a mask file, which will have those CID information, those spot X and Y coordinates. And then you need to have a reference data set. For most of the model organism you have the reference, but if we don't have a model organism or any kind of reference data set, then one has to create that. And then we need that image to use that so that you can lay over the RNA on the image. So how does it work? This is a standard pipeline. You take the reference data, FASTQ file, and the CID. You do the mapping of all your FASTQ data sets to the reference genome. And then you merge all the barcodes to create a count. And once the count matrix is created, you do the annotations, you do the reduplication based on the UMI data. And here you register that image. When you register that image, it has track lines. And those track lines tells you where are the spots located. And based on that track line, you can lay over those RNA on the image itself. And if you have multiple tissues, or if your tissue is smaller than your chip size, we need to cut that tissue. And if you have multiple tissue, again, you have to cut those tissues. So separate those tissues as well and do the downstream analysis here. And you can do all kind of downstream analysis because it's really a very generic FASTQ file data set with and then you get a gene count matrix with UMI and X and Y coordinate as separate columns. We have some capability of looking at the data using StereoPy, Python. You can do some kind of visualization here, or you can do some cloud-based visualization here as well. But as I said, this data is humongous. I would think that people will do their own in-house lot of analysis. They will do third-party algorithms. They will develop their own algorithms to analyze the data. Now let's look at some of the advantages of Stereo-seq. Now, Stereo-seq has a sub-cellular resolution at single cell segmentation you can do based on the nucleus staining. You have a way to do, like there are programs like Spatially, there are programs, there are other kind of programs like Stereo-seq, which uses just nucleus staining and then either through watershed or using centroid as a way of figuring out how do you segment the cell. So here, in this mouse embryo, which was published in 2022, each of the cell type is depicted by color, and you can actually do cell segmentation for each of the cell. Now imagine you have a cell of 10 micron here, which is typically a size of a mammalian cell, but of course sometimes you have macrophages which could be going 60 micron as well. A 10 micron cell can have 400 spots because, as I said, between 2 spots we have 500 nanometer distance. So in this 10 micron whole of this area, you can have 400 spots. You can merge all the 400 spots and you can get good number of genes and transcripts. The second more important advantage is field of view. You can put a whole 1 centimeter by 1 centimeter tissue here. Actually, not one. We like 0 by 0.9, so that some of the track lines can be visible. The recent paper in Cell had is 3 centimeter by 5 centimeter. They put the monkey here, the whole of macaque. They have put it. The paper is out in Cell now. The other paper, which was in another Cell paper, the one which I was showing you, they used 2 centimeter by 3.5 centimeter. So the idea is that in one snapshot, can you look at the whole of tissue and then look all the aspects of that particular tissue or whatever is the objective of that study. Now, this is a comparison of one of the technologies. I'm sure you can figure out which one versus Stereo-seq. And you can definitely see a huge resolution in Stereo-seq versus any other technology we can look at right now. Now let's look at some of the applications of it. Right now, so this technology is only 2 years old, and you can see a lot of papers are already out. Some of them are in bioRxiv, and I need to update this graph actually. Some of them have recently come again from bioRxiv to the main paper, published one. So that's something which we think that will come in next year, a flood of it. And on top of it, you can see a lot of papers which are talking about only the Stereo-seq algorithms, or how do you analyze the spatial transcriptomic data set. Not only Stereo-seq kind of data set, but other kind of data set as well. And all these papers have Stereo-seq data set used to figure out what is the best way of analyzing this kind of voluminous data sets. Now, one of the papers I thought I'll discuss here is spatial resolution of axolotl brain. I picked up this paper because here they are looking at the normal developmental of the brain and a regeneration of brain when you create a wound in this brain. And the very first thing what they did here is they created a map, a cell map of axolotl at the cellular level. Two things very interesting here. The first thing is in this particular brain, which has, for example, dorsal pallium, medial pallium, lateral pallium. Then you have all those septum and other areas. If you look at closely, you will see that different cell types occupy a particular space. So it's a very distinct way of cell type occupying the spaces. They're not really kind of mingling much. The another interesting thing is that inhibitory neuron, which people thought that probably they will be in one particular area, they were dispersed in this particular medial pallium. Here you can see that, which is marked by SSTs. SST is one of the markers to pick up inhibitory neuron, and they are all throughout this particular region, which we call it medial pallium. And this was very remarkable in the sense that nobody thought that if you look at the cellular region at a subcellular or at each cell level, how is the picture? And this looks like a very clean picture of cell types being aggregated or clusters in a spatial way. Now let's look at the developmental biology of it. So they took at different time points based on the length of the axolotl. They took at ST14, and the length decide what is the stage of the salamander or axolotl. And they took at ST54, ST57, juvenile, adult, and then the metamorphosized one. What was remarkable here is that this developmental related radial glial cells, which are blue in color here, they persist till stage 57. And the moment it comes to juvenile stage, it just vanishes. It literally doesn't have much of it. Very small percentage you will see that. And that's a very distinct movement from a developmental movement from 57 to juvenile. And then it continues to be like that. And of course, there has been papers earlier talking about this glial cells. Probably it has stemness, and they tend to become neurons. And this might be the way that they have become neurons over a period of time. Another interesting aspect, they look at also the stemness and the proliferation activities of this developmental radial glial cells. And if the stemness module, the cell cycle module, which is actually the proliferation module here, and the translational module, they persist. These are very high here. If you look at this level, these first three are up to stage 57. And then after this is juvenile and then adult module stages. The first three, you see a huge, this module expressions are huge. And suddenly after that, they just kind of diminish. And again, the hypothesis they have put forward is that they become neuronal cells and they lose stemness and other aspects of it at juvenile. Now, compared to that, they wanted to look at what happens in the regeneration. So in this particular graph, so they created a wound in one of the hemisphere. And this particular, all these photographs are like 2 days post-injury, 5 days post-injury, and so on and so forth, up to 60 days post-injury. And the colors are all of different cell types. And when they created a wound, at the 2 days post-injury, they started looking at this red color you can see here, which is much more distinct here. They call it as reactive radial glial cells. And they were a different kind of glial cells, not the normal developmental glial cells. All this here, red ones. Till 20 days post-injury, they persist. But as 30 days post-injury, they do not have those glial cells anymore, those reactive glial cells. They just vanish. Moreover, the ones which had nothing, you don't see those reactive glial cells at all. They are not there. This is only on that part of the hemisphere where the wound is created. That's very interesting thing they observed here. The second interesting thing they observed was these radial glial cells, which they call reactive ones, has some of those markers, gene markers like NES, KRT, TNT, and CCN, NA, which indicates stem cell activities and high proliferation of these cells. They were very high on only this particular cells, those reactive red ones, which are here. You can see that, this photograph. So based on this, they found out that at 20 DPI day post-injury, after that, these reactive glial cells, they just vanish, and their stemness and whatever is there, they persist till 20 DPI. However, in the next one, what they are looking here is, okay, if those are persisting till 20 DPIs, what are they doing? Two modules are upregulated quite a bit. The first module is the wound module, which starts very early. And the second module is the proliferation module, which actually follows the first module of wound. You see, in the reactive ones, the red ones, you have a very high expression level of all those modules very early, and they start decreasing. Whereas the proliferation module follows that as a second wave, it keeps on going up. And the non-reactive ones, the other RGCs, they do not show any such kind of modules up regulated. And you can see here at 2 days post-injury up till 20 days post-injury. The first one, those green ones are these wound ones. They persist till 10 DPIs, and then it kind of is followed by the proliferation activities of those cells. Now the question was, what happens after 20 DPIs? Okay, you had wound taken care of, then you have a proliferation activities taken care of. So what's the next stage happening here? So they started looking for transitional ones. They picked up another new kind of cell, which they call immature proliferating cells, reactive immature proliferating cells. These reactive RGCs move, they kind of transforms into RAPCs, and then immature, a subtype of neuronal cells, and then mature neuronal cells. So these are the stages from reactive, it moves to NPTX after 20 post-day injury. And it beautifully shows here that those which are RERGCs here, somewhere here, and with the velocity curve, they are trying to show it here. The reactive ones are, the immature ones are here, which finally becomes the NPTX ones. And then I think they use monocle as well to showcase that at the pseudotime level, how reactive radial glial cells become N2PX. And also at the same time, at the pseudo-time level, those gene expression of the stemness and other kind of markers, they start diminishing over a period of time. So ultimately, so in a nutshell, if you look at what is happening at the developmental stage, you have a neuronal progenitor in the radial glial cells. They become immature neuron and then mature neuron. Whereas in the regeneration in the wound, you have a reactive neuronal progenitor, which we just discussed, becoming a RIPC, the intermediate progenitor cells, and then you have immature neuron and the mature neuron. And there are two very distinct ways of developmental happening. I mean, one is a normal development, another is a development post-injury. This is I just picked up this paper because I just wanted to focus how the developmental disease can be looked through a model organism. So in this embryo, there's a mouse embryo, they took different sections, 53 sections from stages, embryo stage 9.5 all the way to 16.5. And they created a nice 3D model of spatial transcriptomics showing a very dynamic mouse organogenesis at a fine cellular level. They also picked up a lot of unidentified cell types and cell states both. Now, if you look at this particular two diseases, which are very common, not common, I mean, these are genetic diseases in human. One is called Robinow syndrome, which happens when there is a mutation in Wnt5a. And a patient usually have a developmental problem in maxilla and limb. And here in the embryo, they are trying to show that the Wnt5a is a gene expression quite a bit. It is localized in the maxilla and limb. And probably that's why if you have a mutation at Wnt5a, you have a problem in maxilla and limb. The other one they picked up was NKX21. If you have a mutation in NKX21 in human, you have brain lung thyroid syndrome. And patients usually suffer from a severe lung problem, a respiratory problem. And then you have problems in this part of the area called hypothalamus. And in the embryo of mouse, they show it very clearly that this gene expression is localized in hypothalamus and lung. And this is one way of looking at how the disease can be picked up at a single cell level or in the spatial context here. This is one of my favorite papers, actually. So what they did in this paper is, this is a paper where they took two different kinds of liver cancer, and they took multiple regions in that liver cancer. They took the tumor core, they took the invasive margin, and they took and they also had paratumor sections. On top of it, they also had lymph node normal and lymph node tumor as well. And they did both single cell and spatial transcriptomic, and they're quite a bit focused on the margin area, and they're very, in a very creative way, they analyze this margin area, and ultimately they validated a lot of their stuff using ISCs and RNA protein data, and some of them using mouse data as well. So if you look at different sections they picked up from the liver cancer, the first thing they were looking at, how are those cell types spatially in let's say in a tumor or in the margin area of one of the tumor type, in the margin area of other tumor type, how they are spread out, how they are located. And for example, in the margin area of ICC, you can see a lot of B cells sitting here, the immune cells sitting here, a lot of fibroblasts sitting here. And then they created for each of the tissue a cell density graph here. Each of the peak corresponds to one of the cell types here, and the distribution. They have not shown, I mean, I've not shown here. They also picked up the tertiary lymphoid structure, TLS. And the idea was to look at this TLS, how far and close they are to the tumor core in all of the tissue types. And you can do this kind of exercise because you have nice spatial information. Yeah, and this is a little bit of complicated graph analysis they have done. So this is a margin. You look at this margin here. Now this margin, they have tried to make a schematic diagram here. Now this margin has two directions. One is along the margin, what they call tangential direction. So you're going all the way from this part of the margin to all the way this part of the margin. And then you have another direction, which is going away from the margin, either on the tumor side or you are moving towards the paratumor side. They call it normal direction. Now they did a binning on the normal direction. So they asked, okay, this is my zero. This is a zero data point. I move at a particular distance. So the binning was based on the distance on the paratumor side and on the normal side, on the tumor side. So they did a binning of 0 to 250 micrometer on the tumor side, and then 500 to 250 micrometer further, and so on and so forth. A similar kind of binning they did on this side. So you did on this layer, then you can do on the second layer, on the third layer, moving from the border towards the paratumor side or on the tumor side. And based on that, the simple thing they asked was fraction of cell types. How much of cell type do you see when you move away from the border area? And what they saw remarkably was that all the immune cell type was extremely, extremely high on the border area. In this, particularly in this 250 to 0 micrometer towards the border, but towards the tumor side, and suddenly drops just outside the border of this invasive margin. And then they continuously focused on this kind of analysis, which I'm going to show you further. The next question they were asking, okay, if I figure out that there are immune cells are there, a lot of fibroblasts are there on the margin area, what are the pathways which are enriched here? So some of the pathways which are very enriched on the margin tumor area, not on the margin paratumor, not on the tumor ones, are like, for example, glycolysis, hypoxia, oxidative phosphorylation. All those things were very, very high on the margin tumor area. And so they concluded by saying that that region, this very close to the margin on the tumor side, are characterized by hypoxic microenvironment, robust inflammatory response. As I said, a lot of immune cells are there, and high immune scape as well. So this immune scape, some of the modules are very high, which will clear in the next slide, actually. So here they were looking again. So what you do here for the same beans of the distances, the distance bean, you are looking for those gene expression modules. And of course, you get GSVA score, typical in the single cell people do. So the question they were asking is, what are those modules which are upregulated on the border or close to the borders? So they found out that some of those, like, which are very high, were proliferation capacity ones, CMT ones, and some of the other genes as well. And the ones which were away from the tumor were very high on this kind of pathways like fatty TCA cycles and all those stuff. This is another paper where they use a chemotherapy, and they were looking at what happens in the chemo after chemotherapy when you have a complete responder versus non-responder. So they looked into all those who had complete responder and who had no response at all for this chemotherapy on rectal cancer. So they picked up seven patients which had complete response versus 5 patients which had no response on the rectal cancer. And then they did single cell RNA-seq data and they did stereo-seq data. A lot of stereo-seq data they tried to validate using single cell data as well on this. And quite a bit focus was on the tumor microenvironment in this. So the first thing they wanted to know is how are the, if you're looking at tumor microenvironment, you look at the CAFs for sure, all the fibroblasts for sure. And they were looking at what kind of fibroblasts you see in the in this complete responders versus the NRs, no responders. Let me call that CR versus NR. So they found that CAF had certain very distinct kind of profile with the gene expression like PI16, SLEET2, ADM28. They are quite a bit on CR compared to NR, whereas bone morphogenic protein 4 or fibroblast associated protein or MFP1, they are very high on those non-responder ones. And they were also looking at how they are distributed across. And you will see some of the interesting things based on the distribution as well. And all these things, they ultimately kind of validated using this single cell RNA-seq data, this particular information. Of course, they cannot validate the spatial context. Now, one of the important gene is prostaglandin. This gene has a role here in rectal cancer. So they were like trying to look at what happens to these syntheses and their receptor. So the syntheses are on the endothelial cell vein, and the receptor of this particular PG gene is on the calf. So they wanted to look at whether what is happening. So they saw a lot of synthesis with EC vein on the EC vein and a lot of receptor on the calf on the complete responder CR1s. The remarkable thing is that a lot of them are co-localized here on the CR1s, and they're totally missing on the NR1s. They do not see much of localization at all. And they do not see this only in one tissue type. They have more CR versus NR, and they have a very similar kind of profile on theirs. And this kind of analysis generates more of hypothesis to do further analysis on the interactions and all the stuff. Another thing they wanted to look, as I said, not how they are looking at the calf and the surrounding areas. So the question they were asking on the responder versus the negative responders NR, what are the immune cells surrounding the calves in a 3-bean range? I mean, you can do the beaning by yourself, like how many spots you want to take, 3 spots, 4 spots, 5 spots, and that's how the beaning can be done. It was very interesting that dendritic cells were quite high on those calves which are in the complete responders one, or let's say CD8 were very high. They were much lower in the non-responder ones. Here you can see that. And that was another very interesting thing they picked up here. Not much on the B cell though. On plasma cell, you see kind of, it's there on both the sides, but these particular CD8 ones and the dendritic cells were clearly much more on the CR versus NR. I'll skip this one. There's a similar kind of study they did. The other one they were looking at, how is the calf with this FAP expression and the malignant cells, their co-aggregation or co-localization. And if you look at the NR, there are a lot. This tumor cell are in this color, whereas this calf with FAP, you can see all of them are like really together, and it's totally missing in the CR1. Again, this is one tissue, one sample we're talking about, but if you look at other samples, they have a very similar profile. That calf and tumor cells are co-localized, where this is missing here. And then they do a very systematic analysis as well, and you see a very flat, there's nothing co-localization, whereas you see co-localization on the responder side, non-responder side. This is the non-responder, this is responder one. I just wanted to, if somebody is interested in other kind of model organism like Drosophila, this was where they created a 3D image on the embryo and larva, which is not easy to work on. They took every third section of the entire embryo and larva, and they had, I guess, 15 total sections, and then they created a whole of 3D image of based on the spatial transcriptomics. And this is going to be my last slide, because this is interesting because we have a ligand receptors here interacting with each other. In zebrafish, they were looking at NOS2 ligand receptors. What they found was the ligand receptors are far away, the interaction is much lesser, but over a period of time, given the same distance, the interactions become much more dynamic, and it happens much more than we would expect early in the stage of zebrafish embryogenesis. So NOS2 receptors in this, over a period of time, they keep on interacting much more than in the early stage of embryogenesis. I'll stop here, and I'll be very happy to take any question.
We have 2 questions, 3 questions now, posted on the Q&A on the Zoom. The first question is, can you speak a little more about the Stereo-seq analysis platform? Is it free, and how do we access it?
Yeah, it's free, and I can help you to I can help you to find out how to access it, but it is free. You might have to use for the computational thing, in the sense that if it is on cloud, then you might have to pay to the cloud, whichever cloud you're using. But yeah, it's free.
And we have a question from Maria. what is the minimum RIN requirement for tissue quality to get meaningful data? And what is the cost of bioinformatics per sample?
What's the cost of analysis per sample?
Bioinformatics.
So the RIN is, we love if the RIN is 7 and above. I can swear on that. If you have 7 and above, you get amazing quality of data. But on the human side, usually we get 5+, 6+, and we accept that as well. Sometimes in the surgery, let's say if you're doing an oncology, somebody is there with a surgeon and they just take the resected tissue and put in nitrogen. The RIN is usually very good. But 5+ and above, we have seen that RIN is very good. And we have done the correlation between a number of genes and the RIN is extremely, extremely high. So we always say, please try to have very good RIN because these are very important samples you're going to use here. Cost of analysis per sample, I'm not sure how to quantify that.
Let me answer. So come back to the RIN. So what we do is if it's above 7, we say, because what happens here, we ask you to QC it before shipping it to Durham, then we ship it over, then we QC it there, see if it matches up. And if it's below 7, we'll reach out to you and say, you know, it's 6 or 5.5. Do you want to go forward with it? If you're comfortable, we'll run it, but then you'd have to pay for it on that. And above 7, we'll just let you know that it passed QC, and then we're going to move forward with the project on that. So that's the RIN question. So it's QC'd on both sides. So we make sure we got, you know, a quality sample. The price then, so the average sales price here in North America that we're getting right now, that's per chip. And it's the 1 centimeter by 1 centimeter. I know Dr. Kumar had a lot of other ones up there, but that's the ones that's commercially available right now. And so depending on the size of the sample, you know, you can get maybe 2 or 3, but if it's a bigger sample, you get 1 on there. And the chip price stays the same. So average sales price right now is about $10,400 per chip here in the US. So if you get 3 samples on there, it's still the 10,000, whatever. We got very special prices for Beth Israel. So considerably less, especially for the end of this year and going in the beginning of next year. So you got my contact information a little bit there, and contact me and kind of talk about the project and all that. The pricing. And that price is everything. So we get the sample there. We do all the slicing, the preparation, the 2 steps. It includes primary and secondary analysis. So FASTQ file, you usually, you know, try to, if you got AWS, upload it from Singapore. We got a secured site in Singapore. It's not an internet, it's an Ethernet. So it goes up in our bucket, comes over here. Or if you need hard drives, there's different ways of transferring. But it's a lot of data. The 1 by 1 centimeter, you're looking at about 1.5 billion reads. So it's a lot of data that comes.
Right. I mean, just to add to what Craig has said, theoretically speaking, in 1 centimeter by 1 centimeter, if you have a very uniform cell density, you can have almost like 1 million cells because that's how it is. With assuming that a mammalian cell is 10 micron, and which means if you're doing 1 billion even, it's 10,000 reads per cell, much lower than what you do in 10x or any other, whether you use 50,000 reads. We always say, okay, you want to use 1 billion, go ahead and then see the data. If you see it is meaningful, please top it up based on how much cells we are able to pick up.
And it's not biased, it's not role-based. So
yeah, because CDN is very stable.
Yeah.
And just to clarify, the $10,000 per chip, that's all the QC, the running the chip. Does it include bioinformatics?
So it includes up to gene count metrics, which you can use for in downstream because we won't know what kind of analysis we plan to do. Yeah.
Okay. So the output would be a gene count Excel sheet.
Right.
Yeah.
I mean, won't be an Excel sheet.
Okay,
it's humongous data, but it's going to be a
CSV,
gene count metrics.
CSV,
Yeah,
CSV,
it's a CSV.
CSV.
Yeah.
I open it in Excel, but CSV file.
But we have, so then we also do complex or tertiary as well for some. You guys have great bioinformatics.
Especially for the pharma companies.
Some organizations that need bioinformatics, we can, do by, I think we charge by the day, and it's pretty good team. Some people say, well, we want to purchase like 2 days and kind of train the trainers. They got people new that are kind of doing this. So it's all over, or we will do we do projects outside of this. We do bioinformatics projects alone. So we have our data, then we have the capacity to get done. We have bioinformatics to projects as well, just like bioinformatics. But this is, the same. So it's a lot of data. But pretty much, if we hand everything to you, you got a bioinformatics team. It's great.
Thank you, Craig. One more question. It's Maria again, and she says, you talk about the advantages of Stereo-seq, which sounds great. And are there any potential disadvantages or limits of this technology compared to the other currently available genome-wide spatial transcriptomic approaches such as the Visium?
Right. It's never nice to say the disadvantages, but yeah, FFP we cannot do right now. We're trying hard, but it is a random hexamer one, and I think Visium also uses random hexamer. But we do not know when we are coming out, maybe next year, FFP. The other thing which was a disadvantage, probably is no more anymore disadvantage, is earlier we used to do the H&E section and then we take the adjacent one. Now we can do on H&E directly. So that was an advantage. So the protocol is out, commercially available now. Take the H&E section, put on that. So advantage of using H&E is that your cell segmentation will be very much better because your nucleus is already kind of stained, and then your cytoplasm is also stained in H&E. It helps you do a much better cell segmentation. I imagine somebody will be much more creative that they will add at least 1 or 2 cell membrane stains as well, but not a single cell membrane can stain all these cell types. But if you put 2, let's say in our lab, since H&E is a bright field, we can add 2 more cell membrane stains, so up to 3 colors, let's say. But there are no algorithms available which can do H&E plus cell membrane stain together. That will be awesome. I'm sure somebody will pick up this idea that let's have a cell segmentation informed with nucleus, cell membrane staining, and cytoplasm staining. That will be one of the best way of cell segmentation. But I don't think CellPose is one algorithm that uses a lot of H&E data, and it's a very good algorithm for cell segmentation, but it doesn't require cell membrane staining.
Thank you, Dr. Kumar. Is there any other questions from in person? Anyone here has questions? No? Okay, yeah, so that's great. That was all the questions we had. Oh, sorry. And my screen.
I mean, I have a question that's maybe a little out there. Would you ever consider expanding the spatial technology to DNA rather than RNA?
Right.
This would be of interest in cancer, where we're trying to understand, patients. Intratumoral heterogeneity,
Right.
And other structural variants.
So we don't have on the spatial, but we have a single cell whole genome and whole exome, which first set of data is out, and the data looks very good. Because typically in single cell whole genome amplification, you have a problem of amplification bias, huge amplification bias, and then you don't tend to cover the entire genome. With a 15x depth of coverage per cell, we are able to cover 15x depth of coverage. We are able to cover almost 80% of the whole genome, but we also have a whole exome. Coupled with that, on the same cell, you can also assay the RNAs, DNA and RNAs together. So somebody is looking at some kind of interterminal heterogeneity on the copy numbers, or even on the truncal mutation and subclones.
Yeah.
So using some single cell WGS or WES, one can potentially look at it along with the RNAs as well.
Yeah.
It's also special, or
No, that's not special, unfortunately, right now. Yeah.
So how would you proceed to do such analysis? as such, how is it workflow before an experiment? You would dissociate your tumor.
You will dissociate the tumor and then using some kind of a marker, or say, let's say you want to pick up tumors, because I would imagine that you will pick up tumors, those tumor cells, depending on whether it's epithelial tumor cells or connective tissue tumor cells, or brain cells like astrocytes or whatever, depending on what cancer type. The first paper got out in Nature actually on that technology we are using, where they picked up between Alzheimer's disease versus the control cells. And they showed very clearly that Alzheimer's disease have very different kind of signatures at the DNA level. All the neurons don't divide, but then they get damaged and oxidations kind of thing. And then they pick up mutations, and those mutations have a very distinct signature over the other cell types.
Is the 15x depth sufficient then also diagnose structural variation?
Yeah, I think for structure variation, I would say 15x might be even overkill. If you look, it might be an overkill.
So it's good enough.
It's good enough. Yeah, 15x is good enough, and you can pick up even single nucleotide variant. It will be quite a bit computationally heavy lifting of the analysis part, because imagine you are doing 100 cells, 15x. It's like doing 100 human genome, 15x, and then you have to joint call them so that you get very nice mutations. And always do bulk. If you do bulk using some other technology like a normal technology, then you remove all the germlines. Whatever is left is either error or the somatic mutations. And there are nice programs now like Scan2, Potato. Scan2 is out. The Nature paper is Scan2, and the other one is Potato. Potato is much easier to use in the sense that it takes much lower amount of time. It's computationally. Scan2 takes ages to remove the errors, and they use all kind of machine learning. I think Potato use random forest to pick up errors, and the sensitivity is almost close to 86%. So yeah, and then once you have those mutations, then you can create your own clones, subclones, and all the downstream analysis. And if you use RNA as well, then you can ask question, how is the RNA impacted, or the group of RNA impacted based on your clones on the same set of cells. So that would be a much interesting way of looking at. But unfortunately, we are not doing at spatial level. We are not doing at spatial level. But I imagine that if you are doing stereo-seq RNA, and then you can anchor that with the single cell, because you will know the RNA profiles very similar.
Yeah.
And then you can probably do similar kind of studies.
So if non-mutation frequency is below 5%, then may not be detected, right? Because for only 15 coverage. If
But
below,
you see, there are
Single cells.
but that's a single cell. You're looking at 2 chromosomes. I mean, just one set of chromosome in per cell. So a somatic mutation, clonal somatic mutation, will have the same haplotype across all the cells which have the same clone, right? So you can basically do a haplotype caller, not even mutate to pick up. It will behave like as if it is in same haplotypes, and you can very nicely pick it up. And that's a big advantage of using single cell. The other technologies like Melbeck, MDA, Lianti, all those kind of ones, or Picoplex, they always suffer from amplification bias, GC biases. This one, and there are a lot of error rates. The one we are using is something called primary template, PTA, we call it the primary template amplification, where once a copy is generated using PCR, that copy is not anymore amplified. You always amplify from the primary template. So the PCR-induced error rates are not propagated that much because you are always sequencing the primary template all the time. Yeah, hopefully, if she invites me next time, I'm going to give you single cell whole genome and on the stem cell RNA technology. We have generated quite a few on the tissues, different tissues and cell types. The data looks very good. And we have
This data that you just showed us looks amazing.
Yeah.
It's robust.
So the single cell whole genome sequencing is
whole exome.
whole exome sequencing is it's a new thing that you guys just established,
Yep,
right?
yep. And RNA along with that.
And RNA along with that.
On the stem cell. Yeah.
Thank you.
Thank you.
Gaurav.
Yeah.
Okay, so it's about time. No last questions. I'm going to wrap this up. Thank you so much for coming. I really appreciate your time. And for those who join us virtually, I also appreciate you for joining us this morning. Any questions? you can reach out to Dr. Kumar or Craig if you are around the country and you are interested in this technology. Reach out to Craig, and if you are at BIDMC, you can reach out to Craig or me. Doesn't matter. Craig and I talk a lot. Okay, thank you. Thanks everybody. Bye.
Thank you.
Thank you.